Everyone. Good afternoon to the webinar, our informational webinar for our current funding opportunity for Discovery Foundation Awards. I just want to make a couple of quick announcements before we get going here. Yes, thank you. So this webinar pertains to the Foundation Awards that are due February 6, 2025, and that's an important distinction as we've had a previous webinar on the Foundation Awards previously, and there are some changes to this current cycle that we need to make you aware of. One change is that this is going to be the final Foundation Award cycle, and CIRM will announce new discovery stage funding opportunities in response to recommendations from our governing board going forward. So February 6th is going to be the last opportunity to apply for this specific funding opportunity. Following the webinar, we will hold a question and answer session that will focus on your general questions about this program. If you do have questions that are related to your project or a proposal that you're thinking of submitting, please submit those types of questions or request a consultation at our email address, which is discovery@cirm.ca.gov, and that email address will appear again at the end of this webinar if you didn't catch that. So we began all presentations at CIRM by stating our mission, which is accelerating world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. And of course, all of our funding opportunities are in support of this mission. Just a quick introduction to some of the panelists that you will be hearing from on the webinar today or during the following session on question and answer. Myself, my fellow team members, Dr. Uta Grieshammer, Dr. Chanlek Tan, Dr. Janie Byram, and Dr. Haley Lamb will be speaking during the webinar in different portions. And then we have Mira Sarwal from Grants Management, who will be here to answer any questions specifically related to grants management. And I want to extend special gratitude to Ruby Karimji, who is our new project manager, who is running the webinar behind the scenes and making sure all of it goes slowly, and she will be helping to moderate the questions. So the agenda for our webinar today is as follows. In the first part of it, we will be going over the presentation. You'll be hearing about some of the updates and an overview of the DISC0 program, including some of the new elements that were introduced in this cycle, including a second track, which is for multidisciplinary collaborative teams, in addition to the single PI track, which is what you might have experienced if you've applied to DISC0 awards previously. Dr. Haley Lamb will then go over the review process and criteria, and then we will hand things over to Janie Byram, who will talk about application components and considerations, who will then hand things off to Uta Grieshammer, who will speak about some of the information and resources that are available to you. And then we'll come back to me for some common issues. And then we will all go into the question and answer session where you'll have an opportunity. So with that, I would like to hand things over to Dr. Chanlek Tram, who will tell you about some updates to the DISC0 program and provide a general overview for you. Hey, Chan?
Great. Thanks, Kelly. On the next slide. So we'll take this chance to sort of help clarify some of the understanding of the PA without, we try to avoid reading the PA to you. Just a reminder that please go into the PA itself to read more about the award. So I want to give a broad overview of what we are looking for in this award. If you've applied to our award before, this is the same as previous cycles. The DISC0 award is our basic science award. It's focused on foundational and mechanistic research, addressing bottlenecks and addressing knowledge gaps and bottlenecks. So the goal is to get a novel biological insight or tools and technologies. So we list 5 main categories for the proposed objectives here. These are not mutually exclusive, but it's helpful, I think, to have a brief discussion on each of these here. The first is the understanding of the basic biology of stem and progenitor cells. So everything about the basic biology of stem cells and progenitor cells, including differentiation, reprogramming, cell biology, the effects of stress and aging, etc. The second is to advance the application of stem cells in basic research. So this includes advancements in stem cell-derived models, organoids, assembloids, and bioengineering solutions, technologies that are applied to stem cell modeling to answer foundational questions about human biology, whether it's functional genomics or imaging or other modalities that are applied in conjunction with stem cells. and as well as advancing the application to model and study diseases in ways that are not possible with other models. The third is to advance the application of stem cells in regenerative medicine, so which includes cell and gene therapy. In our understanding of gene therapy, this includes RNA-based therapeutics as well. So, for example, we could look into factors that contribute to functional to the function of stem cells in the therapeutic context, persistence, survival of transplants, immunogenicity, and other factors. The third is to address bottlenecks more generally in regenerative medicine, including cell and gene therapy. So for example, these could include better understanding the properties of gene therapy vectors, gene editing effectors, etc. And finally, we are also interested in proposals that extend the reach of regenerative medicine to diverse patient groups. For example, understand the effects of ancestral diversity or the effects of sex on regenerative medicine. So I think that's it. I think just to note as well, we have no limitations on disease indication or organ system. And we do value projects with translational potential, although we ask that you include projects that are early stage and basic discovery. So on the next slide, we're going to address the minimum criteria. These are our scientific eligibility criteria for the proposal to move on to review. So as I explained, this award is really focused on knowledge gaps. So firstly, we need you to identify and address in a basic knowledge gap or bottleneck related to stem cells or to regenerative medicine. Your proposal should be centered on human biology. So we do allow the use of non-human models to be included as supportive studies, provided that you justify and try to attempt to validate within the award period using using human models, tissue, or data. And finally, this is not exactly an eligibility criteria per se, but the vast majority of applications do include human stem cells, although we do keep the door open for applications that do not use human stem cells, but apply genetic research that is relevant to cell and gene therapy more broadly. So if you have questions around that, do feel free to contact us and schedule a consult. Next slide. So as Kelly mentioned, we do have a significant change this year. Instead of the usual single PI tract, that is $1.5 million in direct costs for 3 years. We have added a new team track, which has a maximum budget of $3 million, again, for a duration of 3 years. In terms of the applicant team, the single PI tract does not require any co-investigators. You should avoid listing collaborators as co-investigators if you're applying for the single PI tract. But for the team track, in addition to the contact as a principal investigator, we require that you have a minimum of 1 and a maximum of 2 co-investigators. And the minimum % effort devoted to this project for the PI and the co-Is for either of these tracks is listed here at 15% for the PI and 10% for the co-I. And again, this award is open to both academic, nonprofit, and for-profit research institutions. Next. So one common question we've seen with the registration is, and the consults that we've had so far is, which track should I apply to? For conventional proposals, projects with discrete objectives that can be achievable under the leadership of a single investigator, we do recommend that you stick to the single PI tract. For the team track, this is really to support multidisciplinary collaborations of 2 to 3 investigators that can benefit from a unique integration of diverse disciplines. So if you are if you have 2 or 3 PIs that are essentially working in the same domain, applying the same techniques, and just benefiting from additional scale, that's probably not what we're looking for here. So we have included here the relevant review criteria that you can find on page 10 of the PA, which lists how we would go about judging the merit of a team track proposal. So the reviewers are asked, do the proposed collaborations, particularly for these team track projects, offer a unique synergy or advantage that augments the potential impact of the project? So if you've clearly determined that that's a yes for you, then and you can benefit from the additional scope of the higher budget, then that would be the appropriate track for you. Next. So the next page is on the investigator eligibility. So both the PI and the co-Is of the team track must be employees of a California-based organization, and should not have any similar overlapping activities pending other CIRM opportunities, which is not relevant since no other CIRM opportunities are available right now. For the principal investigator, there is only one PI that can be nominated for all applications. They must commit at least 15% effort to the project. And as a PI, you cannot submit more than one DISC0 application in this cycle of any track. For co-investigators, again, we ask you to reserve this designation only if you are applying to the team track application. If you have collaborators or other key personnel in your project, please just list them as collaborators. Again, the co-investigators must commit at least 10% effort to the project. They should not be part of the same lab as the PI. We should understand them to be independent, have an independent research agenda. And for projects that have a substantial omics data generation, we do require that you nominate a data project manager. If you do not currently have a data project manager for this role, you can just put TBD, but that's something that we will follow up with you on projects if you are awarded. Yeah, next. So this is just an illustrative list of activities that we will fund through this award. The first 5 is basically recapitulating the earlier slide on scope, but it has some additional types of projects or activities that we do commonly see in our DISC0 proposals. So tech and tool development related to the use of stem cells or applying to stem cell modeling, the study of human tissues as ground truth for stem cell models, for example, atlases or other kinds of big data projects, as well as target and biomarker discovery related to regenerative medicine or reverse translation related to regenerative medicine. In most of these cases, we do ask you to either bring in a stem cell component to your proposed studies or make that the relevance to regenerative medicines, basically including cell and gene therapy, quite explicit. Next. So these are the list of out-of-scope activities which DISC0 will not fund. The first one is a very common question that we get. There is this understanding that CIRM looks for projects that have high translational potential, and that is true. So we do like projects or questions that have value as for translation in the future. But in this particular award, it is really targeted to basic discovery and knowledge gaps. So if you have activities that are really towards optimizing a therapeutic approach or selecting a development candidate, that would not be appropriate for this award. Other translational, preclinical, and clinical stage activities would, of course, clearly not be eligible in this award as well. Projects that devote most of their budget to deriving new stem cell lines is not appropriate for this award. This is not a biobanking award. and activities that are already budgeted or for prior existing or future CIRM awards are also not fundable. Next. All right, I'm going to pass it off to my colleague, Haley Lam, to tell you more about the review process.
Hello, good afternoon. Then thanks. I will take over a little bit here for a few slides to talk about the review process. So just so that you understand what happens to applications after they're submitted to CIRM, we do internal checks for the eligibility items that Chan has just gone over, as well as everything that's covered within the PA announcement, just to make sure that the applications are complete and it can be reviewed by our scientific panel. The applications are then accepted for review, and for the DISC0 program, we're anticipating to use our two-part review, which includes a positive selection step and then the full merit review by the panel. Those applications that go to the full merit review then get a grants working group score and recommendation, and those applications are then sent to our application review subcommittee of the board to make final funding decisions. So to go into a little bit more detail on this positive selection step, because it's important for applicants to understand, can you go to the next slide, please? So for our programs that have a large number of applications that come in, it's not possible for our panel of 15 scientific reviewers to critique all of them in detail. So we go through what we call positive selection, where they conduct a pre-review of all the applications to identify essentially a subset of them that are most impactful and most responsive to the program, meeting the goals of the program, and which Chan has already outlined for you. And then those applications that are selected then go to the second stage of review, in which the individual applications get critiques and summaries back to the applicants. So therefore, what that means is that if your application only does not progress past stage 1, for example, is not selected to go to stage 2, those applications essentially do not receive any specific feedback from the scientific panel. So just so that you understand. And it's also important, and I believe Janie will be covering this in the application process, to pay attention to those areas in the application that the reviewers will be focusing on for that first stage, because those are the key areas that they'll be looking at first for that first stage. Next slide, please. So if your application is selected and goes to the full scientific review, these are the review criteria. They haven't changed in the big picture in terms of what the questions are from the last round. However, they have changed in the details. So please very carefully review the criteria sub-questions that are outlined in the PA. I do want to point out that this little asterisk here, the assessment of the potential for impact of the project has changed from prior DISC0 rounds to incorporate the ideas and the synergies that could result from the project because of collaborations. And in particular, that would apply, of course, for the team track projects, but also could apply, it's also a review criteria that is considered for all submissions, including the individual sort of applications as well. So I don't want to belabor the point, but just to point out, there were some adjustments on the review criteria based on the change to the program. And then finally, to just point out that if you are interested in seeing what has been funded and in the past, and in terms of what the feedback has been from applications that have proceeded to that second stage of review, we do have posted all of the review summaries from the prior DISC0 rounds. So please take advantage of looking and seeing the types of projects that and the feedback from the scientific review panels in our website. Thank you. And I will pass it, I believe, to Janie.
Thanks, Haley. Next slide, please. I'll go over the application components. So to get started with an application, you'll log into our grants portal at grants.cirm.ca.gov. You'll select Open Programs to start a new application, and then select Start a DISC0 grant application. Next slide. So there are two main components to the application, an online section and an upload section. And in the online section, you'll complete responses to prompts regarding eligibility, personnel, the project, including the positive selection preview page, funding, which includes budget justification and certifications. And then in the upload section, you'll find the templates that are required to complete the proposal, the timeline, the biosketches for key personnel, other support documents for the PI, and if you're submitting a team track application, the COIs as well. And for-profit applicants, the solvency worksheet and documentation. And as you complete, as you add to the application, the sections on the application navigation panel will change from incomplete to complete. Next slide, great. Positive selection, okay, so you've already heard a little bit about positive selection, and this is a part of our two-stage review. So the positive selection preview page provides the primary description of the application that's available to reviewers in the first phase of the review process. This section focuses on significance and impact and should sufficiently describe the key aspects of the project, as reviewers may primarily use this page to make their determinations in the first phase of review. So we encourage applicants to devote significant attention to the completion of this page. Next slide. The budget. In the funding section, there are subsections for entering subcontract information, co-funding, direct and indirect project costs, as well as a budget justification. So applicants will add their annual direct and indirect project costs in the funds calculator, and subcontracts and co-funding amounts will be pulled into the funds calculator from a separate section. And then the applicant will provide a rationale for the costs per year in the budget justification subsection. Next slide. Uploads. The upload section of the application has templates for the proposal, project timeline, biosketches for key personnel, and other support. These uploads are required. They have a red asterisk next to them. There is an option to upload letters of support and IP licenses and MTA documents if desired. All documents should be uploaded as a PDF, except for the project timeline, which is an Excel spreadsheet. Next slide, please. For-profit applicants must complete the financial feasibility assessment and solvency form and provide solvency supporting documentation. So it's required that you use these provided templates that are in the upload section. For-profit companies must have 180 days cash on hand at the time of application to demonstrate financial solvency. Do's and don'ts with the proposal template. Please use the current required word template. Please adhere to the prescribed page limits and maintain the formatting that includes fonts and margins. Please follow the instructions for each section and convert to a PDF when finished to upload. Please do not alter the margins or font size, etc. Do not exceed the page limits. Do not leave sections incomplete. And please don't use your own template or DIY template. To resubmit applications, okay. If you've previously submitted a DISC0 application and it did not pass the first stage of review, positive selection, meaning you did not receive a review summary or merit score, then you will submit it as a new application. If you are submitting a team track application, you will submit it as a new application. There are no resubmissions for the team track because it's a new track. If your previous DISC0 did pass positive selection in one of the two most recent cycles, and you received a merit score, then you'll select yes to indicate a resubmission on the positive selection preview section. And you can also submit a resubmission statement in the proposal template. So applications are due on February 6th, 2025 at 2 p.m. Pacific time. For programmatic questions, please contact discovery@cirm.ca.gov. And for review questions, you may contact review@cirm.ca.gov. And for budgeting and grants administration questions, please contact grantsmanagement@cirm.ca.gov. And this webinar is being recorded, and the slide deck and also the FAQ will be posted on our Funding Opportunities for Discovery Stage Research webpage in about a week. Okay, so I'll go over a couple pieces of information and resources for applicants. And I'll start, let's go to the next slide. Thanks. And so I'll start with our data sharing requirement, and then I'll pass it off to Uta, who will go over our human embryo and embryo model research in California, as well as our IPSC repository and also resources available through the shared resources labs for stem cell-based modeling. All right. CIRM data sharing requirement. So to maximize the value of CIRM-funded data, CIRM has a data sharing requirement, and that means that if awarded, you will agree to work with CIRM to develop a data sharing and management plan, and that by the end of the award, you will deposit applicable data into an appropriate data repository. And those are just some examples of acceptable repositories. And briefly, there are 3 elements to a data sharing and management plan. There's a metadata catalog, which details the metadata associated with your experiments, a data use limitations or DUL's institutional certification, and that details any restrictions on sharing data generated from biospecimens that you use, and a questionnaire that on anticipated data sharing activities. And so the DSMP is intended to guide the management of your metadata throughout the award, such that when it's time to deposit your raw and processed data in a repository, you're organized and prepared and not surprised by the effort that's required to share your data. Next slide, please. So CIRM is launching an online platform called CIRM Data Explorer to enhance the findability of shared data. It will be a public dashboard for CIRM-funded datasets, and metadata about completed datasets will be publicly searchable and linked to the deposited data in established repositories. And this is also the platform where awardees will submit and update their DSMP. So this schematic shows the workflow. At the beginning of your award, you will submit an initial metadata catalog and data use limitations form, as well as a questionnaire. And then throughout the award, you'll update your metadata catalog and, if necessary, your DUL. And this all happens in the CIRM Data Explorer. And by award close, you will have deposited your data and metadata in an appropriate repository. And the metadata about your project will then be publicly searchable in the dashboard on CIRM Data Explorer. Next slide, please. And so on the CIRM website, you can find more information about our data sharing guidelines and our data repository guidance. And this is under For Researchers on the Information for Applicants page. And there's more information on, as I said, the data sharing guidance and appropriate data repositories. So now I will pass it over to Dr. Uta Griesheimer, who will cover the additional resources for applicants.
Thanks, Janie, and good afternoon. So as Janie mentioned, I will have one more piece of information that's particularly relevant for DISC0 awards related to embryo and embryo model research. So research using human embryos has made major advances in recent years, and so including the successful culture of intact embryos longer than previously possible. So I'd like to remind you first of the regulations in California that are cited here, both the CIRM regulations, as well as the California Department of Public Health guidelines for human stem cell research prohibit the culture in vitro of any intact human embryo after the appearance of the primitive streak or after 12 days, whichever is earlier. Most people are familiar with this limitation in California. Next click, please. So there have been also, as you all know, major advances in modeling human embryos using human stem cells. And there has been some uncertainty how these regulations shown above may or may not apply. So I'd like you to be aware that CIRM's Standards Working Group, a group of experts that makes recommendations about scientific, medical, and ethical standards pertaining to CIRM-funded research, that they issued a guidance on the oversight of human stem cell-derived embryo models that mimic peri- and post-implantation stages of development. And CIRM recommends that the protocols involving such models be subject to annual SCRO review and approval, that the reason for the use of the models shall be included in the scientific rationale, and that the experimental stopping point should be defined ahead of time and not be open-ended. And so the slide deck will be made available, and you should be able to find these guidelines here on our website, and you can also ask me if you can find them. Okay, next slide. I'd now like to make you aware of 2 resources that are relevant to the DISC0 opportunity. About a decade ago, CIRM funded the derivation of roughly 2,600 human-induced pluripotent stem cell lines representing the diseases listed on the right here. The cell donors were patients, mostly with complex idiopathic disease, although some monogenic conditions are represented as well. And of course, unaffected donors were also included as controls. So the repository housing these stem cell lines is managed by FCDI, and you can find information about these lines and how to order them at the address that's listed at the bottom of the slide. Importantly, the tissue donors represent diverse ancestries, and some other useful features include the fact that the tissue donors were consented based on a standard consent template, and that the derivation and quality control of the lines was standardized. There are pre-negotiated license agreements available for commercial use, and there is a SNP genotyping data available on almost all these cell lines, and whole genome sequencing data on 299 of them. Next slide, please. Another resource we'd like you to know about are the shared resource labs for stem cell-based modeling. Those CIRM-funded core facilities that are launching right now, their purpose is to support California researchers and educators by providing access to core facilities, specialized services and equipment, by sharing their models and their expertise and resources broadly across California, by training researchers in using these models, and by providing educational resources and activity, including formal stem cell techniques courses. Next slide. There are 11 shared resource labs across California. They are opening for business, some soon, some later. I'd like to emphasize again that these core facilities are open to researchers across California, not just to investigators at the hosting institutions. We are developing a website where you will be able to find details about these offerings in these core facilities. But in the meantime, please contact me if you are interested in making use of these offerings as part of your application, or if you know of researchers who may be interested, and please do let them know about it. Yeah, thanks. I'm not 2 more slides that I will not go over, but this is just, I'd like you to just be aware. Once you get access to the slide deck, I wanted you to be aware about the expertise that is being shared of 2D cultures and organoids representing a variety of cell types. And the shared resource labs also offer access to technologies and services to support the creation and the analysis of stem cell-based models. And next slide. Almost all shared resource labs offer a formal stem cell techniques course and other educational opportunities. So for now, please contact me with any questions, and we'll announce through our listserv when the shared resource labs website on the CIRM website is up and running. And I'm going to hand it over to Kelly again.
Great. I just unmuted myself. Thank you, Uta. Now I'd just like to spend a couple of minutes going some of the most common issues that we see with applications, so you can try to avoid some of these errors. Okay, first of all, I'll go over some common issues we see in the eligibility screen. So one really common issue is a proposal that is focused on animal biology rather than human biology, or missing the human cell validation studies that we mentioned. So Chan, when he presented, he mentioned that it is possible to use and supportive animal studies and sometimes studies of animal cells in parallel with human. However, there's an expectation that any discovery made in an animal cell or animal stem cell should be validated by using a human cell counterpart for that. So primarily, this is an opportunity for investigating human biology. But I just want to make the clarification, it doesn't completely rule out use of animal tissues or cells, just that the human cell validation part is important. Another common issue is when the proposal is not framed around filling a knowledge gap or overcoming a bottleneck, even if the activities proposed are allowable. If the proposal reads as too product focused, as in we've already discovered a therapeutic approach and we want to optimize it so we can take it into translation, that's not what this opportunity is about. This is really around basic biology, foundational research, testing novel hypotheses. And of course, studying mechanisms involved with therapeutic approaches is fine. It's just that you want the proposal to be rather exploratory and not really, really product focused. And by product, we mean a therapeutic or even a commercializable tool. So that proposals that are too product focused may be more aligned with some of our later stage programs, such as DISC0, which are the Quest Awards, or our translational and clinical stage programs that you may have remembered from the past few years. Another common issue is not using the required templates that are provided for uploads. Those are absolutely required. And if the percent effort of the principal investigator or the co-investigators is below the stated minimum in the RFA, that will be an eligibility flag. Now, in addition to eligibility issues, we are also commonly asked about what affects merit, which is the score that is applied by our grants working group or scientific peer group. So often in this particular funding opportunity, one common weakness is if the proposal seems too exploratory. So if there is not enough preliminary data to convince reviewers that the rationale is worth exploring, or if there is a likelihood of achieving enough result during the duration of the grant that an impact would be made, it might be considered too exploratory. Poorly responsive, that means that there is a lack of a convincing link to stem cells or regenerative medicine. So there should be an effort to tie whatever you're studying to the CIRM's core mission of supporting stem cell research and regenerative medicine, genetic research into human biology. The feasibility is another common area where grants working group might identify weaknesses. So they can perceive risks in experimental design. This could include, for example, if you propose a study that's highly dependent on the creation of a complex tool that does not yet exist, reviewers might question whether the proposal is feasible if there are any issues or delays in creating that tool. Another common feasibility issue is missing expertise on the team, or if there is uncertain access to necessary tools or resources in order to complete the program. And some of that can be mitigated by including letters of support from a consultant for the expertise that might be missing, or somebody who might be providing those tools or resources to you that your project depends on. A poorly written grant application that is difficult to follow can lead to a lower score. Failure to acknowledge relevant existing literature is another issue, and a poor DEI statement or addressing the needs of underserved communities, diversity, equity, inclusion. Please take that section seriously. It is an important consideration in the review criteria. And that concludes the formal part of our presentation. And we are a few minutes early, so we'll be able to take a few additional questions. And I'm going to stop sharing my screen now and hand things back to Ruby Karimji, our project manager, to moderate the questions. Thank you.
Thanks, Kelly. Yeah, we'll jump right into the Q&A, and we'll start with a question that's been frequently asked in both registration and today. So the first one is for Kelly. Are there any focus or priority areas for this round of DISC0?
There are not any specific focus areas as far as disease or tissue or organ. The only things to keep in mind are the linkages to either stem cell biology or regenerative medicine, and to propose foundational studies as were described in the webinar slides and in the program announcement.
Thank you. And to follow up on that, does DISC0 prioritize completely novel research, or is there interest in bringing previously identified targets to clinical applications?
Well, so bringing a previously identified target to a novel clinical application, if if you were proposing, say, here's a therapeutic candidate that was applied for disease X, and I want a study to see whether I can apply it for disease Y. If this is really focused on showing proof of concept and disease modifying activity to go into translation, that would seem to be too product focused, as I was saying earlier. Whereas if you were proposing to explore the basic biology of perhaps how this therapeutic mechanism acts, and then explore whether or not that could be have potential to target other disease pathways, then that could be responsive. So novelty and innovation is always something that reviewers seem to be excited about, but so are studies that have a real chance of leading to something that can eventually impact patients, lead to discoveries of novel biomarkers or targets. So you don't have to have a very innovative proposal in terms of a completely new pathway that nobody's ever studied before, or a completely new tool that nobody's ever looked at before. If you can make an argument that the new elements that you're going to bring about by performing your research in your project is going to have a significant impact and advance the field.
Thanks, Kelly. Our next question will be for Jan. How much preliminary data is required for the application? Are there any stipulations for this?
There are no formal stipulations, but you should remember that this is quite a competitive grant. So we do expect there is that you will have some data supporting the feasibility of your strategy and the significance of your question being asked. Yeah, and before I go on, and sorry to just hijack this because I see a number of questions related to co-Is and PIs and team tracks. So I just wanted to take this opportunity to kind of clarify our expectations here. If you're applying for a single PI track, do not list any co-Is. You can have as many key personnel as you like. Collaborators, you can list them as key personnel or collaborators, and these could include people that are outside of California. If you're applying for a team track, you must have a minimum of 1 and a maximum of 2 co-investigators, and those need to be California-based. Everyone else should be listed as collaborators or key personnel. And we do not use the term co-PI here, so don't use that as well. So thanks.
Thanks, Shin. And our next question will be for Haley. Can PIs submit more than one application? And second part to that question, can a PI submit as a single PI, but also as part of a team track from the same institution?
Right. So I think just to piggyback a little bit off of Chan's answer just now as well, an applicant can only submit one application as a PI. However, you can be a co-I on a team track application as well. And I don't believe we have a formal limit on like I guess you could, I suppose, be a co-I on multiple team track applications. But the limitation is one PI application.
Great, thanks. And another question for you is, do the resubmission policies for grants between 80 to 84 still apply?
Yes. So if you had a application for DISC0 in the prior round that received a score of 80 to 84, that application will bypass the positive selection step and will go straight to full scientific review.
Okay, our next question will be for Mira. Does the split of funding between PIs have to be equal?
Thanks, Ruby. The split of funding between co-investigators does not necessarily need to be split equally. Typically, the distribution of funding among co-investigators would depend on the proposed budget based on the specific roles, responsibilities, and contributions outlined in the grant proposal.
Great, thanks. And then for Janie, can someone serve as both a co-I and the data project manager?
Yes, that's possible. Whether a project has substantial omics and requires a data project manager will be evaluated after the funding decision has been made and during the pre-funding administrative review. So the COI can also be the data project manager, but if you're also in a situation where you're unsure whether you'll require a data project manager or who that would be, you can put TBD in the application.
Great. And then another question for you is, where will the data be deposited, and can a non-CIRM collaborator access the data after the ending of the project?
So your data will be deposited in an established repository that the applicant identifies. CIRM has provided data repositories guidance, which is available on the CIRM website under Information for Applicants. And what will be available to be searchable by the public at the end of the award is the metadata that is associated with your data sharing and management plan, and that will be searchable on CIRM Data Explorer, which will be a new dashboard for CIRM. We will send out an announcement when that is launched. And a non-CIRM collaborator can access the data after the ending of the projects. Can you repeat that question again?
Yeah, can a non-CIRM collaborator access the data after the ending of the project?
So that all depends on the access that you put on the data in the repository. If you have it under controlled access or if it's publicly shared, the PI has the authority on whether their data is shared publicly or not.
Great. And then the next question will be for Uta. Must the proposed human-induced pluripotent stem cell work use clinical grade lines? And if so, is there a supplemental fund to cover the cost?
The short answer is no. This question, in a way, would pertain to our former DISC0 award mechanism, where we did look for researchers to end up at the end of their research project with a newly identified therapeutic candidate. As has been now mentioned, I think several times, that is not the goal of DISC0 award, and clinical grade human pluripotent stem cell lines are not they're definitely not required for this kind of research.
Great. And then I see, Kelly, you're going to answer a question live. It's that, do you have to be a CIRM grant awardee to attend the workshops and courses?
I think you're talking about the shared resource laboratory offerings that Uta mentioned, and many of them are going to be available to the public and other institutions. So no, you do not have to be a CIRM grantee to benefit from the resources offered at the shared resource laboratories.
Great, thank you. And then the next question will be for Haley. Will CIRM provide peer review feedback on submitted grant proposals?
So peer review feedback is provided for those applications that receive the full scientific review, meaning they have been selected in stage 1 of positive selection and proceed to stage 2. Those applications that are not selected in the positive selection process do not receive any peer feedback.
Great, thank you. And a question for Kelly. What is the success rate for grants funded by DISC0?
So we have had three prior rounds of DISC0 before this one. And typically the funding rate has been on the low end, it was more like 10%, and on the high end, it was about 17 or 18%. So average is about 12 to 14% if you average across the three.
Great. And then another question is, can you explain the difference between direct facilities costs and indirect costs?
Is that for grants management, Mira?
Yeah, that could be for grants management.
Sure. So I think I can answer this question best by just kind of going over how facilities and indirect costs are calculated. So nonprofit applicants can request facilities costs for research awards consistent with their current federally negotiated rates when available. When research is conducted off-site, a grantee may request a portion of their facilities cost based on their actual out-of-pocket lease costs. Currently, indirect costs are capped at 20% for most awards. Exceptions include, other sort of award mechanisms like training and conference programs. But these rates are applied to the adjusted project costs, which exclude equipment, tuition and fees, research, patient care costs, etc. And the total cost of each service contract, subcontract and consultant agreements in excess of $25,000. And to build off of that, for-profit applicants may request facilities costs up to 35% of the adjusted project costs and are not eligible for indirect costs. So for organizations that do not have a federal rate agreement, internal documentation supporting both the allowable facilities cost rate and the methodology used to arrive at the amounts requested in the application are required at the time of contracting, so at a later point. And I'd be happy to go over more, information pertaining to the difference between facilities and indirect costs, specifically facilities B costs, maybe at a later point or separately outside of this public forum.
Great, thank you. And then another question for you, Mira, is do you have any insight on how this application may differ from NIH funding?
Yeah, that's a great question. So while there are many commonalities, a few key differences can include California-specific eligibility requirements for both the applicant institution and the PI, various IP and independent publication rights, which must be held by the parent institution in California. Out-of-state subcontractors or consultants cannot retain those rights. And so any costs of activities performed by a separate out-of-state organization that does retain intellectual property or independent publication rights in any intellectual property, such as invention, technology, data, arising out of CIRM-funded project funding is unallowable. To add to that, other personnel don't necessarily need to be named specifically in the application. And then probably of most importance, there is direct project cost caps that do apply to the application's budget. So subcontracts and indirect costs associated with some subcontracts can apply towards the direct projects cap, which is different than the NIH's practice in application budgets. And then just to add, outside of the application, but worth noting, a key difference in how no-cost extensions are reviewed is worth highlighting because NIH generally lets awardees extend a funded project up to 12 months without submitting a prior approval request, while CIRM requires a prior approval with a clear justification to be submitted. And usually the no-cost extension period is up to 6 months, and specifically in this award mechanism, that is the case.
Thank you, Mira. And then going off of the budget question and topic, this might be a question for Kelly. Is there an estimate on how many grants will be awarded during this cycle, and also how many will be funded through either the single or team track?
Yeah, this is the first time we've offered a team track, and so we're uncertain of how many applications we're going to get of each type. But based on historical precedent and the planned budget allocation, we expect that will probably be about 12 awards made. 12 awards made that would support half team track and half single track. It could support more if it were all single track and fewer if it were all team track.
Great. And then for Haley, how much change in scope can be implemented in resubmission?
So I think that's a pretty hard question to answer, and it's project specific. I think the important thing is that an applicant is responsive to the prior review, whatever those comments were. The reviewers do remember the applications that have come through previously and are looking for applicants that have, taken into serious consideration the critiques from the prior submission. So
Great, thank you. And another question for you, Haley, would be, what is the timeline of review? And can an overlapping proposal be under review at another grant agency?
So I can't remember the exact timelines, but it's about a 4-month cycle, I believe. So the eligibility, positive selection, and all of that, you would not be notified of necessarily of your review results for several months. I believe the times are in the PA. And what was your second half of your question? It's escaped me.
One second, I'm trying to find it as well. I think we might have clicked it so it's gone. Oh wait, and can an overlapping proposal be under review at another grant agency?
No.
Great, thank you. So I see that we only have about 1 more minute for this session. I'll just ask one more question, and for anyone who didn't get their question from either registration or today answered, we will be putting together an FAQ. And just another reminder that if you aren't sure if your project is the right fit or have specific questions related to your research, then you can email us. And the slide deck and recordings will be made available on our website. So for a last question, let's do, can instructors on a non-tenure track position be PIs on single track awards? And Kelly, would you be able to answer that one?
Yes, so it depends on your institution if they are willing to stand you up as a PI. The PI is responsible for as the point of contact and for all of the reporting requirements to CIRM and ensuring compliance. And the institution would need to support that individual as a PI. If they otherwise can lead a project and the institution is able to support that and sign off on their application, then that individual could be named as the PI on the application.
Thank you, Kelly.
Mhm.
And with that, that's the end of our Discovery Foundations Award webinar. Thank you everyone for joining.